Okay, sorry for that. And it's my pleasure now to introduce the plenary speaker for session 17, Dr. Iddya Karunasagar, who is Senior Director of International Relations at NIT University in Bangalore. And Dr. Karunasagar has extensive background in the area of microbiology, toxins, harmful algal blooms, and seafood safety. And from 2007 to 2014, he was Senior Fisheries and Industry Officer at FAO headquarters in Rome, where he focused on food safety and quality. And he has been involved in numerous efforts related to understanding and assessing impact of biotoxin on mollusks. And today we'll be presenting on climate change, harmful algal blooms, and health risk in One Health context. So please, come. So you need to.
Okay, thank you. Thank you very much for the kind introduction and good morning to all of you. I would like to thank the organizers for the kind invitation to be present here and make this presentation to you this morning. Now the topic is on harmful algal blooms and health risks in One Health context. Can I have the full screen slide? So just to refresh what we are saying about One Health. Now this recognizes that human health, animal health, and environment are very intimately related. And when we think of reducing disease risks, we have to consider all these aspects. Now, do we have any information on the effect of climate change and human health? Of course, we don't have information on the illnesses that are caused due to, or we can attribute to climate change, but we have some information on the loss of life due to mainly extreme weather events. For example, in India in 2016, more than 2,000 deaths occurred. And financial losses were about 21 billion, which is 1% of India's GDP. And the number of these type of extreme weather events have been increasing. Now, to look at harmful algal blooms, what do they cause? Of course, they cause number of problems. They cause fish kills. They cause shellfish toxicities, and also fish toxicities like ciguatera fish poisoning. And these could be influenced by increase in sea surface temperature, increased nutrient inflow due to heavy rains or storms and hurricanes. Now, what type of information do we have on the effect of these factors on harmful algal blooms? So these were summarized very nicely in this paper by Wells et al. And you can see here, we have effects and arrows going up and down, and the plus sign indicates the level of confidence that we can have. And the effect of different factors like temperature, stratification, ocean acidification, cultural eutrophication, grazing, on different types of algal blooms, diatoms, toxic flagellates, benthic dinoflagellates, algae associated with fish killing, high biomass algae, cyanobacteria. So there can be varied effects, sometimes going up, sometimes going down. So we need to understand the complexity of these interactions between the factors that we associate with climate change and these harmful algal blooms. But what we now realize that the frequency of these type of extreme weather events have been increasing. Here there are some pictures. One relates to the incident that occurred this year in India and also in Middle East. So these type of events are increasing in frequency. And what is the effect on the fish safety? Now this picture from the website of Woods Hole Institute of Oceanography shows the incidence of PSP, say for example in 1970, and what is the situation in 2015. Because in India also we had few outbreaks and we were involved in investigating this. So part of this may be due to increased capacity to detect, improved technologies, but there are also increased frequency of occurring of these type of shellfish toxicities in different parts of the world. Now, the various aspects related to bivalve mollusk safety and this monitoring of harmful algal blooms related to this has been summarized in this FAO publication, which follows the expert meeting that was organized by FAO and WHO, following request from the Codex Alimentarius Commission. So this summarizes the science related to most of these harmful algae which are associated with biotoxin risks in bivalve mollusks. And Codex has also come up with standards for these biotoxins associated with bivalves. But we have more problems with respect to ciguatera fish poisoning. I would like to talk a little bit more on this. Now, as we know, this is caused by consumption of fish from coral reef area that contain toxins produced by the dinoflagellate Gambia discus species. There are several species. And over 400 fish species can be carriers of this ciguatoxin, but common ones are red snapper, reef cod, grouper, barracuda, kingfish, trevally, parrotfish, etc. Now, this is a heat-stable toxin, so it's not destroyed by cooking, and it's highly potent. Ingestion of as little as 0.1 microgram of this toxin can cause illness in humans, and the symptoms usually start between 1 to 48 hours of eating fish. And the symptoms could be gastrointestinal, neurological, as well as cardiological. And this picture again from the website of Woods Hole indicates the area where the prevalence of these toxicities are observed. And generally it was thought that this is confined to area between 35 degrees north to 35 south. But now we see more incidents occurring in different parts of the world. This incident occurred a couple of years ago in Mangalore, in my city. Over 200 people were affected by consumption of fish, which came from one of the processing factories. They ate the head of the fish. And here the ciguatera fish poisoning was confirmed by clinical presentation, the fish species involved, which was red snapper, and also some of the assays like mouse bioassay, receptor binding assay, liquid chromatography, mass spectrophotometry study. But now confirming ciguatera fish poisoning is not easy because the assay is quite complicated, and these algae, they produce two types of toxins, water-soluble toxin and lipid-soluble toxin. The water-soluble mycotoxin is not involved in ciguatera fish poisoning, but it kills mice. So it's important to have effective extraction and cleanup procedures for detection of these toxins. And these functional assays, they will indicate they are not specific, so you cannot confirm unless the liquid chromatography mass spectrometric study is done. So it's complicated. And this figure shows from a paper in 2015 the prevalence of ciguatera fish poisoning in Asia. But now we have reports also from Indonesia, India, so the picture is changing and we have more and more countries experiencing ciguatera fish poisoning. So the geographical range is expanding, now known to occur also in temperate regions of Japan, Southern Australia, Europe. It has been detected in Canary Islands, Madeira, Portugal, Greece, also Gulf Coast. And even in regions where it was known to occur, the frequency has been increasing, as this report from French West Indies indicates, 5 times higher incidence compared to earlier years. And how to manage this? Of course, there are over 400 species which can be involved, both herbivorous as well as carnivorous species. And identifying fish species that carry toxin is important for public health management so that awareness can be created about these fish which could carry the toxin. But there is significant variation in the toxin content between individual fish species between different geographical areas. And we are also encountering new species that are identified as carriers of this toxin. Even shark has been reported to carry this toxin. Now, this toxin is a polyether toxin, and there are three major groups which are differentiated based on the origin as Pacific type, Caribbean type, and Indian Ocean type. And even among these types, there are a number of variations in the molecule congeners of each type. And in Pacific type, there are two types, and these are designated mainly as CTX1 to 4 Pacific type, Caribbean type 1, 2, and Indian Ocean type 1 to 4. They vary in their biological potency. The Pacific type is considered the most potent, being 100-fold more potent than the Caribbean type. And Indian Ocean variants are considered 60% potent compared to the Pacific type. So for management, this is important to know what type of toxin is occurring in the fish. As I mentioned, there are different types of assays like mouse bioassay, neuroblastoma cytotoxicity assay, receptor binding assays, and these functional assays are non-specific. They'll indicate a molecule, say for example, a sodium channel blocker is present, but to confirm the toxin, one has to do an LCMS analysis. But then the problem is the reference standards are not available. And purified toxins are available only in case of Pacific and Caribbean types, and for Indian Ocean type, purified toxins are not available. And acceptable levels of this toxin have not been agreed internationally. Most countries depend on this FDA action level of 0.01 ppb for Pacific toxin and 0.1 ppb for the Caribbean toxin. So the current public health management practice depends on monitoring reef areas for the presence of this toxic species, algal species, but there are more than 100-fold variations in toxicity between species. So we need to know what is the toxicity of the species occurring in a particular area. And also 2 to 9-fold variation within species due to growth conditions. And identification of susceptible species would be of considerable importance, but there can be large variation in the toxicity. Even from a particular location, fish may vary in the toxicity content. And avoidance of large fish more than 2 kg is recommended. Now, when it comes to setting international standards, Codex Alimentarius Commission is the international body setting standards, and they depend on scientific advice, which comes mainly from FAO and WHO. So in this case, now there is action now. The Codex Committee on Contaminants in the 11th session have requested FAO and WHO to perform evaluation of known SIGWA toxins, toxicological assessment as well as exposure assessment, including geographical distribution, rate of illness, congeners, method of detection, and based on this scientific information to come up with guidance on risk management options. So as usual, the procedure in FAO WHO, there has been a call for experts and call for data, and an expert consultation is scheduled this November. So hope we will have more scientific information coming from this expert consultation, which will help Codex Alimentarius Commission to come up with more risk management recommendations. Now I'd like to talk a little bit about bacterial pathogens which may be spreading globally due to ocean currents. Now, Vibrio species are mesophilic bacteria which are associated with warm water environments. Out of these, 3 species are human pathogens, Vibrio cholerae, Parahemolyticus, and Vulnificus. But not all environmental strains are pathogenic. In the case of Vibrio cholerae, there are over 200 serovars, out of which only one O1 and O139 can cause the disease cholera. And among Vibrio parahemolyticus, only 1 to 2% of the environmental strains produce virulence factors that are required to cause illnesses in humans. But these organisms are also associated with a number of marine organisms, crustaceans, fish, algae. They are also present free living in the water. Mollusks are most important carriers of these Vibrio species. Now, particularly Vibrio parahemalticus is of interest because it is occurring in coastal and estuarine environments, global in occurrence, associated with all types of animals, zooplankton, mollusks, crustaceans, shellfish, and finfish. And ecology is influenced by temperature and salinity. And in tropical environments detected throughout the year, provided salinity and other conditions are available. But in temperate climate, detected mostly in summer, and the organisms are overwintering in sediments. And till 90s, this organism was considered mainly tropical, and in international trade, there were even rejections due to mere presence of this organism without considering whether it's pathogenic or non-pathogenic variety. But in 2004, there was an outbreak in Alaska, which is 1,000 kilometers north of the place from where any previous illness was reported. So the range extended 1,000 kilometers with this outbreak in Alaska in 2014. And then this organism is associated with zooplankton. So they adhere to zooplankton, degrade chitin. So chlorophyll may have influence on the ecology of this organism. And studies done in Galicia in Spain indicate that an organism of particular genotype may be extending over 1,500 kilometers in the ocean associated with zooplankton. So this indicates that zooplankton may have a role in the spread of this pathogen. And then in 1996, there was a variety which caused, say, higher incidence of disease, which was belonging to serotype O3K6, which was first detected in India. And then in 1997, disease caused by this variety was detected in Peru, and which spread then southwards along more than 1,500 kilometers till it reached the Chilean city of Antofagasta. And several thousands of cases occurred here in Chile. Now this figure indicates the global spread of this pandemic strain of Vibrio parahemalticus, and you can see some areas where pandemic strains were observed and some where you had Vibrio parahemalticus due to non-pandemic strains. But then how did these pandemic strains spread across the globe? Now molecular studies of the strain suggest that the 1997 El Nino episode provided the corridor for the displacement of this Asian pandemic Vibrio parahemalticus strain to the American continent. And the reference has been given here, Martinez et al. 2016. And this phenomena, El Nino phenomenon, characterized by the arrival of equatorial warm waters in a sequence of waves, and in 1997 it occurred for a period of 6 months. So over a long period of time there has been this invasion of this, what came through El Nino into this coast, and probably Vibrio parahemalticus established there. So this is the paper, one of the early papers where they reported the relation between El Nino and spread of Vibrio parahemalticus to the South American continent. Now this strain, though it was first detected in India in 1996, it was already found from archival strains in Indonesia. So it was occurring in different parts of Asia before that. And the relation between El Nino has been demonstrated in this data, in this figure, which shows relation between the incidence of cases and sea surface temperature anomalies. So the incidence of disease and the arrival of these warm currents are closely related. And similarly, the arrival of Vibrio cholerae into Peru in 1990 is also related to these El Nino events. Now I would like to talk about this Vibrio parahemalticus, which is causing disease in shrimp. So this causes disease not only in humans, but recently we have encountered problems in aquaculture in shrimp. In Asia, there was a major disease problem called acute hepatopancreatic necrosis disease, which caused losses for several billions of dollars to shrimp aquaculture industry. And shrimp production was reduced by 50% in affected countries. And the strain of Vibrio parahemalticus that is causing this shrimp disease is a slightly different variety, and it possesses a mobile genetic element, a 70 kb plasmid, which contains genes encoding the Photorhabdus insect-related toxin. So this emerged in Asia in 2009, first detected in China, then it spread to Vietnam, Thailand, Malaysia, and it has also been detected in the Latin American continent, in Mexico. And shrimp pathogenic strains, luckily so far, lack virulence factors related to human pathogens. So from food safety perspective, they are okay, but since they are mobile genetic elements, we never know when they move to the human pathogenic strains. And interestingly, this plasmid has been found in a number of other Vibrio species, Campbelli, Harvey, Ovensi. Now, what caused the emergence of these pathogens? Could be eutrophic conditions, because following good culture practices, they have been able to control this problem. So maybe the conditions caused by eutrophication caused an enrichment of these type of pathogens carrying this plasmid, which has caused shrimp problem. So we can see relation between animal health, human health, and the plant health. So to summarize, the events related to climate change have caused increased incidence of harmful algal blooms, fish and shellfish toxicities. Geographical range of certain harmful algae is extending, and Ciguatera fish poisoning is occurring in regions not known to be endemic before. El Nino has caused transoceanic spread of bacterial pathogens like Vibrio species, and new pathogens of animals like shrimp, which are related to human pathogens, are emerging due to nutrient-rich conditions in aquaculture environments. Thank you very much.
Thank you very much for this very nice talk. So we have time for a couple of questions. Please remember, you can walk to the microphones. Well, while people are thinking about that question, I have a couple of questions, actually. You were talking about the transoceanic spreading. Do you think that nanoplastic or microplastic could serve as vectors to those Vibrios to infect different regions?
Now, in this case, it has been demonstrated that zooplankton, because it occurs in association with zooplankton, and as the data from Spain indicated, in the ocean, about 1,500 kilometers area, you find the same genotype associated with zooplankton. So it's here, in the case of Vibrio parahemolyticus, it is suspected to be zooplankton, but we don't know about microplastic. We don't have data.
Okay. Anne, please?
Yes, yesterday we heard that in some small communities, aquaculture is sometimes a alternative for food security issues. And yet, your talk has raised several frightening issues associated with the incidence of pathogens in aquaculture. So are there guidelines for small operations?
Yeah. Now, generally, small operations are very extensive, so they don't follow intensive practices. So most of these problems are associated with intensive aquaculture, where they have very high densities of animals, they use a lot of feed, so that causes eutrophication of waters, wastage of feed, and things like that. So most of the problems are associated with the intensification of aquaculture. So there are very little problems associated with small-scale aquaculture, which is generally extensive. They don't feed it, the organisms use natural feed. And even for intensive aquaculture, what is recommended is implementation of good aquaculture practices. So management here in Vietnam and Thailand has been mainly based on adopting good aquaculture practices.
Yes, please.
Yeah. Can you elaborate on the climatic drivers of these changes? So whether it's extreme events, warming, what else? Can you elaborate on that aspect a little bit? Thank you.
Yeah, of course. I think regarding El Nino, I think the next speaker is going to speak about El Nino and its relation to climate change. But I don't know what are you expecting regarding the other.
What kind of drivers are driving these changes? These pathogens are spreading and living more because the waters are warmer, because they have a different composition?
Okay.
You know, that kind of stuff. What is driving this change?
Yeah. Now, in the case of harmful algae, we saw there are a number of factors, like, of course, temperatures, increase of sea surface temperatures, and also the nutrient or changes that are occurring, say, in a storm event or a heavy weather event. So they may be driving the harmful algal bloom events. But with respect, because maybe similar things affect also bacteria, because they are also associated with these plankton. And whenever there are change in plankton populations, there are also a change in these bacterial populations. And some of these are pathogenic, so they are closely related.
Yes.
Thank you. You are telling that that the spread of all these type of microbacteria, bacteria, and for whatever, is mainly due to climate change, to extreme events. But perhaps you should not consider the issue of ballast water. Nowadays, there are more and more boats that take water from China, and once they are in Ecuador or Peru, they discharge the water, and that water contains, all type of of phytoplankton. For example, in Ecuador, we did not have 10 years ago. We did not have some type of this phytoplankton that is dinoflagellate. And Ecuador, for example, and Peru too, receive a lot of water. And regarding cholera, we have to remember the cholera was the first one event of cholera, big event, was in 1917 in Peru, and then some around Ecuador. So ballast water should be considered when we're doing this sort of model or this sort of approaches. Thank you.
Yeah, definitely, I agree that there are a number of other reasons why the bacteria or other pathogens spread. But we need to also consider climate change-related events as one of them. Thank you.
Other question? Yeah, actually I have one. In certain region, is there any predictive capacity of these outbreaks events?
Yeah, I think particularly with respect to Vibrio cholerae, I think there have been some models related to plankton. There have been a number of studies done in Bangladesh where they predicted cholera outbreaks based on these planktonic blooms, because they're associated with plankton. So some work on prediction based on plankton with respect to cholera has been done in Bangladesh, but I am not aware with respect to Vibrio parahemolyticus, we still don't have that kind of information to predict.
Any further question? Well, thank you, Dr. Karunasagar.